All right, thank you very much, Antonio. Thank you very much for the nice, kind invitation also to the other organizers. It's a pleasure to give a talk to tell a bit more about the research we do in Maastricht, the Department of Nutrition and Movement Sciences at the Physiology of Human Nutrition Research Group. So I'm already working as a couple of years as an associate professor within the group, and my research is especially focused on the effects of diet and lifestyle interventions on vascular health. Vascular health assessed both in the periphery and the brain. And that's actually also what I'm going to focus on today a little bit about the work we do and our current research focus. I would like to start with this slide. I think it's pretty important to keep in mind. It's actually stating like the problem we face or we will face in the future. And that's the problem of the aging world population. Now, this slide actually indicates that it's expected that the amount of people over the age of 60, so by definition, older adults, will triple by the year 2100. And why is this important? Because, as you may know, age is the main risk factor for dementia, and it's also, of course, the main driver for age-related conditions. So, based on this slide, you can already imagine that we will face some future problems. Now, this slide indicates the main causes of death in Western countries. And what you can still see on top is cardiovascular disease, so the heart attacks and the strokes, they're still on top. But as you can already see, Alzheimer's and other types of dementia are now already the third cause of death in Western countries. Now, given the fact that the world population will continue to age, we will actually face a future problem, and that's actually the future problem of dementias. And this slide nicely illustrates that the number of people with dementia will triple already by the year 2050. And this actually is especially something we will see in low and middle income countries. So as you see in high income countries, there is a steady increase in the number of people with dementia. But especially in the low and middle income countries, you will see an exponential rise. So probably now it's actually the third cause of death. But if there's nothing going to change, I think dementias will really become the future problem. There is some good news, of course. And the good news is, of course, we can do something about it. Prevention is key. We are capable to prevent age-related conditions. We are actually capable to prevent the development of dementia. What should we do? I think diet is very important. Diet is key. So that's also illustrated on this slide. This is the Dutch Wheel of Five. It's actually from the Netherlands Nutrition Center, actually stating what we should eat, what's healthy food, and what to avoid. And I think this whole thing is already actually mentioned about the importance of dementia years ago by the American inventor Thomas Edison. And he phrased that the doctor of the future will no longer treat human frame with drugs, but rather will cure and prevent diseases with nutrition. I think it's partly true. I think nutrition, I will talk about nutrition today. It's also the key focus of our research group. But I think lifestyle is a better approach. Nutrition, diet is very key to prevent diseases. But of course, also keep in mind it should also be about exercise training, physical activity. So lifestyle factors, that should be the main focus. Diet is key, but keep in mind, diet is only part of the solution. And also, I do not like the word to cure. If we talk about prevention, then, or if we talk about nutrition lifestyle, the key focus should always be prevention. And yes, there are examples that you can really cure with lifestyle, for example, with nutrition as an add-on therapy besides drug treatments. But always keep in mind, if we talk about nutrition lifestyle, prevention should be the main approach and not to cure. A little bit like an overview of the work we do in the Physiology of Human Nutrition group. We used to focus a lot on the effects of diet on the body, on the periphery, the effects of diet on metabolic health, how we call it, in the body. They're pretty well established. And that's actually the left part. So why is this relevant? Because we do know that diet and lifestyle can actually prevent cardiovascular disease by having beneficial effects on metabolism, by having beneficial effects on the health status of the arteries of the vascular system. What's new, or more like a hot topic these days, is to assess these effects in the brain. That we focus still on diet and lifestyle interventions, but that we're not going to assess like solely vascular function in the body, but we're also going to focus, okay, how does this impact the brain? What about the impact of diet on brain vascular function? What about the impact of diet on brain metabolic health? And this is also very important because you can imagine like the connection between brain vascular function, brain metabolic health is way more closely related to cognitive impairment, cognitive decline, and dementia. So the left part is well established, and in our recent studies, we're actually focused more and more to assess these aspects in the human brain to make the connection with cognitive performance. And that's also the key focus of the work in the group right now and of my talk. And I will give today some examples of the work we do. Now, I do not think I need to convince you over here about the relevance of brain health. This is like a high resolution MRI image just showing that the brain is a very, very well perfused organ. There are like many arteries in the human brain. The brain is also one of the most metabolically active organs. So you can already imagine that the health status of the arteries of the vascular system in the brain does impact your risk to develop dementia. That's pretty obvious. The second most important form of dementia is vascular dementia, which is directly related to the health status of the arteries within the brain. And that's the focus of the studies we are actually currently performing. Can we do something with dietary interventions? Can we improve the properties of the arteries within the brain, also in short-term studies? And how does this translate maybe to functional benefits, like, for example, improvements in memory performance? So how do we assess brain vascular health? I think there are many ways to do so. This is, I think, obvious, the most simple way. You just use a camera. So what you can see over here, it's a retinal camera, a fundus camera, and what you simply do, you make a picture of the retina. So that's the nerve layer on the back of the eyeball. And what you just do is you focus on the characteristics of the arteries in the retina. It's a nerve layer, so there might be a link with brain vascular health. Of course, it's very indirect. But the benefit of this approach, it's very easy. So, for example, if you are interested on the effects of lifestyle on vascular health in school children, you can bring these cameras to schools and make these kind of images. So it's a very simple tool. And actually, this is a very old study in which we already showed that with lifestyle, we can actually improve like the characteristics of these arteries in the retina. But again, it's extremely indirect. Yes, it's very simple, but of course, you do not really know yet what's really happening in the brain. Now, a very, very often used tool is transcranial Doppler ultrasounds. So basically, you make use of sound waves with an echo probe placed on the skull. And what you can actually do is you can measure blood flow in the main arteries within the brain. So what you can see over here, not sure if you can see it. So this is so-called circle of Willis. And with transcranial ultrasound, you can actually mainly assess this, the blood flow velocity in the middle cerebral artery. This is a tool very often used, like lots of researchers do state, okay, this is a measure of brain vascular health. I partly agree, mostly disagree. I think it's a measure of perfusion. So you're measuring, okay, what about blood like going to the brain? But still, you do not know what's really happening in the brain. So we do use this technique. I think it's a very simple approach. You can use this approach, for example, during like if you're measuring, for example, memory performance. So you can perform these measurements in real time. So that's a very nice way, but it's still indirect. You still do not know, okay, what's happening in the brain. So pretty obvious. And of course, I do not need to convince the audience over here. If you would like to know what's happening within the brain, we do rely on, for example, MRI techniques. This is pretty nice to see. So the building over here, you all do know. So we're actually in right now. So next to this one, are the Skinexis research facilities, and Skinexis is housing MRI scanners that are mainly used for research purposes. So that's very nice. In our studies, we mainly use the 3 Tesla scanner in our research projects. And the main outcome actually that we're currently actually using is cerebral blood flow. So we measure on, or we focus on the amount of blood actually in different brain regions. For this, we use arterial spin labeling MRI. Arterial spin labeling MRI, what it actually is going to do, you're going to label your own blood when it's actually going through the carotid artery to the brain. And you're going to magnetic label the water molecules in your own blood. So this means it's a non-invasive approach. And then you're going to assess how much of these water molecules are actually distributed throughout the brain. And the colors, they indicate the amount of blood. This technique is often confused with functional MRI. That's not the case. It's not a functional measurement, meaning that we perform these scans in the morning in fasted conditions without any stimuli. So it takes about 10 minutes in the morning, then we make 10 consecutive of these scans, and this is actually the end result. So there are no stimuli involved. It's a resting state scan performed in the fasted conditions. So no triggers are actually involved. Now, why is this important? So as already mentioned, cerebral blood flow is a measure of brain vascular health. It can be used as a measure of regional brain vascular health. And it's, of course, pretty obvious. What we do see if people age is that cerebral blood flow goes down, basically meaning that the blood distribution throughout the brain, there is a reduction if we do age. And therefore, older adults, that's also our main target group. So apparently healthy older adults above the age of 60, those people already have a reduced blood flow. They do have an increased risk for age-related conditions. So that's also our main target group to actually work with, within the context of lifestyle interventions. So this is another slide. This is actually my brain, like I said years ago. Just showing these are actual brain slices, how the end result is actually going to look like. So again, the colors, they indicate the amount of blood in different brain regions. So what you can clearly see, of course, the cortex is highly metabolically active, so it's receiving a very high amount of blood. The white matter regions, they're indicated in blue, so it's a lower blood flow towards these areas. And that's what we actually do in our studies. We perform these scans before and after an intervention, for example. Then we average the perfusion over the group. And then we're going to look like, okay, are there like differences on the whole brain level? Or are we able to detect some regional differences in? Do I admit? All right. Yeah. So as already mentioned, of course, the MRI scans are performed within this building at Skinexis. But please note, besides MRI and brain, we also focus a lot on peripheral health. I do still have a very love for peripheral vascular measurements, vascular function, endothelial function, stiffness measurements. And most of these measurements are performed at the Metabolic Research Unit Maastricht at the University State Single 50 ground floor. We do have a research unit, about 20 rooms, and all these rooms are actually used to assess some kind of metabolic health outcome parameters. And we're using 1 or 2 of these rooms to assess vascular health. So given like the time restriction, I decided, okay, I would like to explain 2 former studies we performed in our group to show you some of like the outcomes we already saw like in these kind of interventions. I would like to start with an older study performed by 2 of the PhDs. I have a double task over here. So performed by 2 of the PhDs, Lea Tischmann, Jordi Kleinloch. They already graduated some years ago. They performed a study on the long-term effects of soy nuts. Why soy nuts? The project was financially supported by the Alpro Foundation, so plant-based nutrition. Soy nuts are a very good source of plant-based proteins, a very good source of antioxidants, phytoestrogens. of unsaturated fats, and they're part of the Wheel of Five. So they actually are part of a healthy diet. And we were interested, okay, soy nut effects are already pretty well established, but the effects on the brain were actually very limited, like reported. So in this study, we focused on 60 grams of soy nuts each day, actually provided for 4 months. And again, like the target group were older adults, so people over the age of 60 with already maybe some like disturbances in the brain. Crossover study, so we requested people to consume these soy nuts for 4 months, or we requested to consume no soy nuts. Now, this is actually the end result. To explain a little bit more detail, this is actually how a CBF map looks like if you're going to average the perfusion over the whole group. So in this study, we have about 25 to 30 participants. So what we did, we asked people to consume soy nuts. And after like the intervention, we made these ASL perfusion scans, and then we averaged the perfusion. And it actually looks like this. So on top, like the blood flow after soy nut consumption, and on bottom, the outcome after the consumption of no soy nuts. Now, just like focusing on the image itself, there are no apparent changes on the whole brain level. And therefore, of course, we always aim to look for regional differences. And that's also what we did in this study. We did some voxel-wise regional or cluster-based analyses, and we actually were capable to pick up 4 brain regions. 4 brain regions, 4 clusters, having a differential cerebral blood flow following the intervention with soy nuts as compared with the consumption of no soy nuts. So I will dig like a little bit further on the location of these clusters in my next slides. In all our studies, of course, we also incorporate functional outcomes to see, okay, do people also feel better or do they perform better? In the project right now, we use CanTab, so we use iPads to assess cognitive performance. And in this particular study, we observed that long-term soy nut consumption was actually able to improve attention and psychomotor speeds. So this is actually the test we performed, in which we actually observed beneficial effects. Now, the main results are actually illustrated on this slide. On the x-axis, you do see the reaction time and movement time. So a faster movement time, of course, it's beneficial. So it means that you're more like you're faster. So that's actually an improvement. And on the y-axis, you see like the increments in blood flow within these 4 brain clusters. Now, these clusters were actually mainly located in the left and right occipital lobe, with some parts in the temporal lobe and also partly in the left frontal lobe. Now, overall, if we look like on the location, we did see that these regions were part of the so-called ventral network. And the ventral network is actually a network known to be involved in psychomotor speed performance. So that's also illustrated on this slide. We were actually able to make an association between the improved performance, so the improved psychomotor speeds, and like the improvement in cerebral blood flow, the measure of brain vascular function, like indicating there might be some relation between these outcomes. And therefore, like the conclusion of this study was that yes, we did observe that longer-term soy nuts can improve brain vascular health. We did pick up some regional differences, and we also could conclude that these effects may indeed underlie the observed beneficial effects on cognition in this case. Now, as a second example, this is a more recent study, a recent study performed by Kevin Ace. Kevin is now working as a postdoc within the group. This was actually the work he did as a PhD student on longer-term consumption of mixed nuts. Mixed nuts, of course, obviously are part of a healthy diet. Mixed nuts are part of the Wheel of Five. So what Kevin actually did, it was a project financially supported by the International Nut and Dried Fruit Council. He actually provided 15 grams of walnuts, 15 grams of pistachio, 15 grams of hazelnut, and 15 grams of cashew each day to the study participants, 60 grams a total. Why mixed nuts? Because mixed nuts, of course, are healthy. They're a very good source of plant-based proteins. They're a very good source of unsaturated fats, both poly and monounsaturated fatty acids, and a very good source of antioxidants, polyphenols. So it's actually a rich mixture of different bioactive ingredients with already well-established effects on the body, on the periphery. So lots of effects on mixed nuts that they're healthy are already known. But like the effects on the brain, they were like less reported. And so therefore we performed this study. We asked people to consume these nuts for 4 months, or we asked people to consume no nuts for 6 months, for 4 months. So this is actually already part of the results. As already mentioned, the effects on the body of mixed nuts are well established. We also did found indicated on the left that total LDL cholesterol concentration, so the more traditional cardiometabolic risk markers improved, which was already reported before. And we also reported effects on arterial health in the body. So endothelial function and stiffness and microvascular function also did improve. The novel thing about this study was to see, okay, do these effects in the periphery also translate basically to the human brain? So the question was, okay, these mixed nuts in the longer term, can they impact brain function and cognition? So again, as already explained, arterial spin labeling MRI after the consumption of mixed nuts for 4 months and after the consumption of no nuts for 4 months. Then a CBF map for one participant looked like this. And if we average all study participants after the consumption of mixed nuts, so that's panel A, and after the consumption of no nuts, panel B, it looks like this. So this is the average. These are actual brain slices, the average blood flow. And again, on the whole brain level, no apparent differences. However, again, as a next step, we aim to focus on some regional differences. And actually, we did. We did observe some regional changes. We observed, or we picked up 3 clusters, 3 brain regions having a higher blood flow following mixed nut consumption as compared with control. Now, interestingly, if we focus a little bit further on the exact location of these clusters, we can see like the green one was more related in prefrontal brain areas, while the blue and the red one were more like part in the precentral and superior frontal brain regions. And why might this be relevant? Because it's known that these brain regions are actually involved in both the retention of verbal memory, but also in visual spatial object recognition, which are actually 2 aspects that are important for memory performance. So our first conclusion was we observed an increased blood flow in frontal and parietal brain regions, which may be relevant for important cognitive processes. Now, cognition was assessed, and like a little bit as expected, we did not observe effects on executive function and psychomotor speeds, but instead, there were 2 areas of memory that were actually beneficially affected. We observed both in the visual effects on visual spatial memory, as the total amount of errors actually went down. This is assessed with the PAL, paired associated learning test. And also verbal recognition memory was improved. So the free word recall actually went up. And therefore, given that the fact that these clusters were actually located in areas that might be involved in like these aspects of memory, we could conclude, like the first conclusion of the study was, okay, we do see improvements in memory, and these effects may be related to the observed beneficial effect on regional brain function. Now, this part, what I'm now going to explain, was a little bit more like exploratory. We observed no beneficial effects on whole body insulin sensitivity. So we performed an oral glucose tolerance test. So peripheral insulin sensitivity was not affected, which already pretty well reported in the literature. But we were more interested, okay, can we also assess the effects of, like the effect of nuts on brain metabolic health? So why is this important? Very brief background, and I think most of you, of course, already know, is that if you consume, of course, a meal and insulin concentrations go up, it's impacting, of course, glucose metabolism, but it's also having an impact on food intake. So what actually happens if you consume a carbohydrate-rich meal is that insulin concentrations go up. And as a result, glucose will be taken up by the peripheral tissues. And also, as a second effect, is that you will see a reduced food intake, reduced consecutive food intake due to the increased insulin concentrations. And we were interested, okay, because we know in some people like these like mechanisms they work properly, but we also do know that some people have some disturbances. They have a disturbed glucose metabolism due to visceral fats, and they have disturbances in and like food intake regulation. So our question was, okay, with mixed nuts, which is a healthy food, which is actually already known to have an impact on satiety, are these mixed nuts actually capable to have an impact of insulin on the brain? And for this, we use nasal insulin spray application. So the question was, okay, with mixed nuts, can mixed nuts actually impact brain insulin action on the brain using a nasal insulin spray application? So very briefly, how do we assess brain insulin responses? It's an indirect measurement, unfortunately, so you cannot measure it directly. So therefore, I do not like the word brain insulin sensitivity. I rather prefer brain insulin action or brain insulin responsiveness. But how do you assess these effects? You first make a resting state or fasting scan of the brain, cerebral blood flow. Then you use nasal insulin and you just spray insulin directly in the nose and it goes to the brain. And after 30 minutes, you make a new scan. And what you're going to do is, okay, you're going to see, are there actually changes in cerebral blood flow in different brain regions? Because these changes, they actually reflect some of the actions of insulin on the human brain. So that's basically what you do. Now, in this study, we observed that the intervention of mixed nuts actually was capable to affect brain insulin action in different brain regions. And as you can see, these regions are mainly located in the occipital brain areas. And why is this important? And that's actually illustrated over here, because we know, see, if you do see changes in cerebral blood flow in these occipital brain regions, that's related to some risk factors. So we do know pretty well from literature and our own review, if you see that there is an increased cerebral blood flow following nasal insulin administration in these occipital brain regions, that's related to a higher age, and that's also related to insulin resistance. What we observed in this study was actually that with the intervention, we were actually able to reduce the action of insulin on these occipital brain areas. And why is this important? And this is pretty like experimental to say it like this. This can be important because these areas are not directly related to cognitive performance, but instead they're more related to food perception and inhibitory control. Now, as already mentioned, nuts are known to have satiating effects. They boost satiety. You can stop the consumption of further foods if you eat nuts. The mechanisms are already pretty established, but what actually unknown was actually like the potential relevance of the insulin action within the brain. So what we observed in this study, and that's actually actually indicated on the right part, is that with mixed nuts, we can actually improve brain insulin action in occipital brain regions. These regions are actually involved in food intake regulation and food inhibition. And we do believe this can be a potential mechanism towards the beneficial effects of healthy whole foods on satiety responses. Unfortunately, satiety was not assessed in this study, and therefore this is actually an outcome that we're actually currently implementing in all studies that are currently running. So the left part I already explained. So actually one of the conclusions of the project was we do see or we do know some peripheral effects of mixed nuts. These effects partly translated to the brain. We did see an improved brain function. We did see an improved memory. And we also provided some evidence of an improved brain insulin action. And this can be especially interesting given that peripheral insulin sensitivity was not affected. So there are stories that brain and peripheral insulin sensitivity always go hand in hand. So provided on these preliminary results, I think you should a little bit be careful about that conclusion, because there can be some differences, although I do believe there is, of course, a relation. So this was actually the end conclusion of this project. So effects on brain vascular function and brain insulin action. This is something we're actually exploring even further in new ongoing projects. So the effects of diet on metabolic health, both in the periphery, but also in the brain. I'm more interested on the left parts, on the vascular effects of these dietary interventions, the link with cognition, age-related conditions. But therefore, I do collaborate a lot with Tanya Adam of our group. Tanya has a main interest in food intake regulation, satiety. She also knows a lot on functional MRI measurements, which we're actually implementing in new studies. So functional MRI bolt scans in response to food cues. And I think because both worlds, they nicely fit in like ongoing work on the health effects of foods on the brain. Finally, I would like to acknowledge the group, the Physiology of Human Nutrition group, for, of course, all their help. And I would like to especially acknowledge Kevin Nace on the right top. Kevin has already mentioned our postdoc in the group. He's continuing doing some very nice, cool work on the effects of diet on brain function. And I would like to especially acknowledge Linda Kerr and Lucia Kerkhoff. They're sitting over there. Two of my PhDs that are continue working, having projects on brain function, effects of diet on brain function. So if you would like to know more about it, Tineke Degens on the right bottom, she's going to join tomorrow, another PhD. If you have like questions, go have a talk with them. I think they can talk with a lots of enthusiasm about their projects, and they can tell you all about it. And I would like to thank you all for, again, for the kind invitation and for your attention.
Thank you. Super interesting talk, and also very much on time.
Thank you.
All right, we can take a few questions from the audience. We have roughly 5 minutes for each speaker, and then obviously there is more time outside. Maybe I can start with one. I wonder if with the CBS there is the opportunity to also look into inter-individual variability in the effects. And if you have done that, also maybe taking into account, I don't know, also some other potential confounding factors, so other sort of background variables, lifestyle sort of changes also in the course of the study. So in general, the question will be how much can you explore this variability, and how much it's usually done in these types of studies?
If you like basically can disentangle the effect of foods from like other variables, for example.
Yes, but also like dissociate whether there are some individuals where you see like super strong sort of positive effects, and others not. And then, okay, can you dissociate if there are some other variables that can be mediating the effects?
Yeah. No, I think a good question, of course. All these studies were like mainly powered on cerebral blood flow. So it was a power calculation done on blood flow in the brain. So basically to make some bold conclusions about cerebral blood flow, brain function and cognition, of course, obviously we need to have the whole group of participants. But of course, we also do some subgroup analyses afterwards to see, okay, are there people that are more responsive and people, for example, that respond less? What's pretty interesting, what you see, for example, in the study with mixed nut, is that people actually having a more proper metabolic health at baseline, they actually responded better, which is pretty like not really that something, because normally you always say, okay, if people are at increased risk, you can see more pronounced effects. Actually, we saw the contrary. So if people are from a metabolic health point of view more healthy, they actually more responded way better. Age is, of course, a very interesting factor. So the age range was pretty limited in these studies, and therefore we are now extending the age from like 45 until 70 to see, okay, do we see, for example, more pronounced effect at a younger age versus at an older age? So we're actually like exploring like really the impact of these characteristics on brain health, like to see, okay, who's actually responding, and if you have some data on that as a next step to see, okay, why and what could be a potential like explanation for that.
Yes. No, super, super interesting. Yes, Nicole?
Thanks for this nice presentation, Peter. I was really intrigued by this brain insulin sensitivity, and especially in the context of all these new hot medicines like Ozempic that are focusing on GLP-1.
Yep.
And do you think there might be a relationship between GLP-1 and the brain insulin sensitivity?
Actually, that's something they're currently investigating. Yeah, so we collaborate a lot with Tübingen University. They actually were the first one like doing these kind of experiments, and they have some ongoing work like now with drug treatment and to see, okay, can you really modulate brain insulin action in these kind of trials? So no data yet. So only data available from lifestyle interventions, but yes, it's actually currently like being investigated in ongoing studies. So I'm really looking forward to see, yes, maybe this can indeed be a very important mechanism, but still there are some disadvantages. So it's not a direct measurement, it's highly indirect. It's a brain insulin action. You do rely on brain regions with insulin receptors. So what about the regions without these insulin receptors? So there are really like very important limitations, but I think like it's giving some evidence about like the story. So therefore, we were really happy to like to see these results, and therefore new studies we all include like brain insulin action and the list of outcome parameters. But yeah, it's currently in the pipeline. Yeah.
Thanks.
Yes.
Mostly related to what you were saying, that as a fact, you've seen that people with better health, metabolic health, are reacting better. Is it possible that nutrition efficiency is a parameter that you have to consider? So the possibility, the actual efficiency in taking the proteins and the nutritional elements from food during metabolism is a key.
Yeah, good question. So your question is basically, okay, are you able to see what's the individual impact of the bioactive ingredients, and are they taken up properly?
Sometimes food goes away without proper absorption. So
good one. Yeah. So the problem is we do have some biomarkers, that's true, but still it's pretty difficult. So we can assess compliance, but it's really difficult to really relate the effects on the brain to really one specific element. What I can state, for example, we do believe proteins can be part of the story, and therefore a new study, actually performed by Lucia, we are actually going to isolate the effects of protein supplementation on brain function and brain insulin sensitivity. So it's not that we only do studies on whole foods, and also by myself, I also have an interest in the individual factors. So therefore, we have some ongoing work on the protein side. We have a project with wild blueberries with the anthocyanins to see the effects of the antioxidants. But if you really would like to know that, then you really need to properly make a study on that ingredient. Because now indeed you do not know really where the effects are coming from. It's the whole food, correct?
And also probably the motility in the stomach, which allows the metabolism, is another parameter that we consider. So the story is probably much more complicated.
I agree. I totally agree. Yeah, thank you. Yeah.
Yes.
Yeah, thank you. That's really interesting. Kind of related to the previous question. Did you look at, well, kind of two parts of the question. In the no nuts intervention or part of the intervention, was there any placebo or anything else that they did? And then kind of second part of the question, did you look at whether, kind of assuming that there wasn't any other intervention, if the nut intervention might have, kind of the additional consumption of nuts, if that might have changed what else they ate? So possibly eating this amount of nuts before the time, did that change? Eating less of some other food. So could it be that the effect is driven not by the addition of nut, but the change in what else they eat, like saturated fats or whatever?
So the answer for your second part, yes, I think so. For the first part of your question, they were requested to consume no nuts during the intervention. I think they properly did. So what's the difference between both? We all asked them to follow healthy diets. So they all followed the Wheel of Five. The only difference was consumption of nuts or no consumption of nuts. And that's actually the strategy we're currently following also in follow-up studies. But for your second part, yes, I think they did, because body weight did not change. if you know, like you provide about 360 kilocalories extra energy each day with nuts, you can already nicely make the calculation by the nutritionist and calculate what will be the weight gain after 4 months. It's significant. Given that the fact there is no weight gain, yes, we do believe they left out, for example, some foods during breakfast, or for example, they left out some snacks. So therefore, yeah, we do think that the effects can partly be attributed to the fact that they left out like some unhealthy foods, for example. But that's the whole concept, basically. You give people healthy foods, and as a result, they're going to leave out some snacks to increase the dietary responses. But that's like the impact of the study. So that was actually already part of the question we had in the beginning.
Did you let
Yeah, we did. We received many questions from reviewers about, okay, what exact like foods did they left out? And with the food frequency questionnaires we used, unfortunately, we do not have a very good overview. Definitely some snacks, that's for sure. But unfortunately, we didn't use diaries, so that's a little bit difficult. It was a questionnaire validated for the intake of nutrients, so therefore it was difficult to really make a bold conclusion. What exactly did they left out? We tried to come up with a answer. Yes, snacks, but if the question is what kind of snacks, it's difficult really to come up with a clear scientific answer.
All right, given the time, thank you very much again, Peter.
Thank you.
You can find me